Hello? Hello?
Preparation of mRNA from DNA molecule.
Right. DNA, mRNA, or not only mRNA, any RNA. Transcription means rRNA, tRNA, any RNA.
Oh,
Means
okay.
RNA. Okay. That is the transcription, the involved enzyme, transcriptase enzyme. Now tell me, what is reverse transcription? Reverse transcription process.
Preparation of mRNA from DNA.
Right. Preparation of DNA from RNA. RNA stand to DNA. So this is reverse transcriptase. The rest of the all process are same. Denaturation, annealing, and extension, they are the same. But only difference is first in reverse transcription because actually what is the genetic material? What is the genetic material coronavirus? Actually, in general, what is the genetic material in organisms? In general,
DNA.
RNA. General
DNA.
DNA is the genetic material almost all organisms. What about viruses?
RNA.
Viruses, not only RNA, not
They
only
contain
DNA, this both may be made up of DNA strands or RNA strand. Anyone existed, anyone, only anyone, either DNA or RNA. If the virus DNA, if the pathogenic virus is DNA, that is this PCR sequence called as real-time PCR. That means one DNA strand prepare more number of DNA. If the virus DNA genome is RNA, virus genome is RNA, the RNA should be transferred or should be changed into DNA. This RNA should be changed into DNA in the reverse process. After that, these DNA copies, we required more and more, we get more and more DNA copies. But same process, same real-time process. But one difference is first RNA should be converted into DNA strand. That's it. Because we can get the more and more copies. Here the main, first of all, tell me the simple answer to me. And which makes number of copies? Which enzyme makes number of DNA copies?
Polymerase.
Right. Which polymerase? DNA polymerase or TAC DNA polymerase? Here TAC DNA polymerase only amplify the DNA strand, not RNA. Remember. The DNA polymerase or TAC DNA polymerase only amplify, only multiply, only DNA strand, not RNA. For we want to get the more and more RNA copies, it is not possible. Only it is possible if it convert into DNA, isn't it?
Yes.
Right?
Yes, sir.
Right. So in first step, clearly given, this way. For example, this is RNA strand. RNA always, it is not a double stranded RNA, it shows the folding, the single strand, but shows folding. First, heat 70 degrees centigrade. Second step, if we take the selected the RNA first should be denatured, right?
Yes.
For this, heat 70 degrees centigrade. If we heat 70 degrees centigrade, these foldings makes a linear, like this linear strand, right? maybe 5 and 2, 3 and or 3 and 2 find any direction. Only one direction. That is only one direction. RNA. Now what would happen? What is the next step?
Uracil should be replaced by thyroid.
What is denaturation? After that, after denaturation, what is the second step?
Annealing.
Annealing. What happened in annealing?
Cooling at lowest temperature. Cooling.
And also complementary DNA strand forms.
Complementary DNA, that means actually, first of all, primer attached at 3n direction.
Yes,
Primer
sir.
attached at 3n
Primer.
direction. This is
Yes.
first the red color RNA. Remember, this is RNA. After attachment of primer, now it forms DNA strand, the blue color DNA strand on the template of RNA. 2, 5 and 2, 3n direction. 5 and 2, 3n direction, right? Now this DNA called as cDNA. This DNA called as cDNA.
Complementary DNA.
Right, complementary DNA. Now this complementary DNA for RNA. Now this both stand. That means RNA stand and DNA stand. Both stand attached with RNA primers. Means, for example, 5 and 2, 3 and right. Five and 2, 3 and right. Okay. Here primer required, it forms another DNA, and here also primer formation 3n direction, and here it forms 3 and 2, 5 and 2, 3n direction, which forms
Lagging strand.
of course lagging or leading strand, but which forms RNA or DNA strand?
RNA. RNA, sir.
How to synthesize RNA? No, actually reverse transcription process, RNA to DNA form. After that, DNA makes DNA number of copies. That is the PCR proper. This is the PCR concept, right?
Yes, sir.
So after get the 3 and 2, 5 direction, one cDNA now attached with primer. Now it's become double stranded DNA, right?
Okay.
Yes.
Now this true DNA form. Now this 2 DNA enter into the cycle. First cycle, second cycle, third cycle. Now we can get more and more copies, right?
Yes.
Is it clear?
Okay,
but initially to get this DNA, what are the changes occur here? First initial stages.
First denaturation followed by annealing.
Right. First of all, denaturation of DNA or RNA.
RNA.
RNA, sir.
Right. RNA denatured. That means separated, appearing to linear. After that, attached with RNA primer, now forms the DNA. This DNA called as cDNA. Now the DNA copies and prepares more and more copies. Now we can identify the pathogen based on the sequence. And we can identify we can determine or diagnosis whether it is positive or negative.
Okay, sir.
Positive or negative identified the based on CT value. Based on CT value, not given anywhere here. CT value means cycle threshold. Cycle threshold. That means, for example, of course, you know, from the RNA, DNA strand prepared from the DNS, and after that, more and more copies, number of DNA are formed. How many cycles are necessary to detect pathogen genome? The total number of cycle called as CT value. For example, let me clear one thing. A is the person, B is the person. A infected with coronavirus and also B infected with coronavirus. After taking genome, that means RNA from the A person and person. After PCR cycle, I mean in thermocycler, after cycles, here from A person, after 20 cycles, get thousands of copies, thousands of DNA copies. But B person, after 35 cycles, get only 200 copies. 200 copies or less than 200. In this both, who is positive or who is negative?
A is positive.
A is positive. A is positive means these more and more genomic pathogen genomes are appear in this person. The same person very less, more or less percentage appear in B in person. That means, of course, he infect with, if during corona pandemic, each and everyone can infect with coronavirus. Due to the immunity power, this virus would be killed. Due to immunity power, would be killed. For example, a person infected with one virus, and the same be also infected with one virus. But this one virus, it is multiple into 2, 4, 5 number of viruses. Within short period. But in the same way, B person, of course, infected with virus, but when it forms into 2 copies, immediately one virus, one particles would be die due to the immunity power of the B person. And next 4 formed, 8 formed, 6 formed, and this multiple number of multiple increasing at the same time, these virus particles would be died due to the immunity power. That means the finally only less variant appear in B person. The same time, of course, infected with one virus, but lack of immunity power, within a short period, 2 becomes 4, 8, 13 number of uncountable number of copies, variants are appeared. That means the number of viruses means number of RNA, isn't it? Hello?
Yes, sir.
Yes, sir.
Now when we separated number of viruses, it forms the number of copies, number of same detect copies, 1000 copies or more than 1000 copies. And after these copies, we can get only 20 cycles, within 20 cycles. That means first cycle 2 and second cycle 4, like this within 20 cycle, more than thousands. That means in first cycle involved, more than 10 viruses involved or 20 viruses involved, because these viruses already existed in a person. The lack of immunity power. Whereas in B person, 1 or 2 viruses are there. These 1 or 2 viruses, these 1 or 2 RNA entered the PCR cycle and forms the just 200 copies only. Just 200 or less than 200 copies. That is the variation. Then how can we find out this genome sequence? The infected genome sequence with help of probe. Probe means actually, is it clear first? Hello?
Yes, sir.
Yes, sir.
Yes.
Right. So this genome sequence recognized with help of the probe. Probe means complementary sequence of the pathogen. The main simple here. For suppose, for our understanding, A, U, G, C. For this is the complementary sequence of the virus. Complementary sequence of the virus. The same sequence attached with probe. Probe should be C for G. G for C. Adenine. Uracil. This is the probe sequence. We know pathogen sequence. We prepare the probe sequence when this both sequence colonized in a medium. These both are attached. Of course, in a medium, number of sequences are there, but only 2 sequences attached. These 2 sequences, based on the sequences recognized, this is the pathogen genome sequence. That is one thing. Another thing, with help of dyes, color dyes, we can detect, recognize the pathogen sequence. So this is the recognition of DNA pathogen sequence. Is it clear?
Yes, sir. Okay, sir.
C2 value here, not given in our textbook. C2 value. Well, remember one thing. Based on the CT value, we can detect the pathogen whether it is positive or negative. More CT value positive, less CT value negative. CT value means, CT value means to detect that, how many cycles are necessary to detect pathogen genome. This total number called as CT value. That means some are taken more than only 20 cycles. For example, another example.
Sir, in the RT-PCR test, if the value is more, then he is much affected. If the value is less, he is less affected.
1000 copies here. Here also same 1000 copies. This 1000 copies formed within the 10 cycles. These 1000 copies are formed 30 cycles. Who is negative? Who is positive?
First one is negative, second one is false. Sorry.
10 cycle positive
10
or 30
cycles
cycles?
positive. 10
Right,
cycles
10 cycle positive. So this 10 cycles number called as CT value. This number called CT value. Cycle threshold. This called CT value. This is also CT value, but the CT value cycles here more. CT value cycles more to formation thousands of copies. If exceed 30 or more than 30 cycles to take detection of the virus, it should be negative. Below 30, it should be positive. That's it. That is the PCR, RT-PCR test mechanism. Right? Is it clear? RT-PCR, real-time PCR, and reverse transcriptase PCR. Okay? Simple real-time PCR, the pathogen is DNA. Pathogen genome DNA. Same process annealing, denaturation and link extension common. Reverse transcription, the pathogen is RNA. Now RNA converted into DNA, then only the same process annealing and extension. So this is PCR and RT-PCR concept. Is it clear? Hello?
Yes, sir.
Clear,
Yes,
sir.
sir.
Okay.
Right. So next one, molecular markers also I will explain later. Because these molecular markers, cloning vectors, they would be covered in practicals. Okay. Next one. Now another easy block, biotechnology, black thought, biotechnology. This is also very, very important. Biotechnology, third unit, 13 unit, introduction to plant tissue culture and in vitro morphogenesis. Turn on page number 193.
Yes, sir.
Just wait. Right. Right. The totipotency. What is totipotency? You must have heard about this. Hello.
Ability of the plant cell to give rise to a whole plant.
Right. The ability to produce a cell, mainly a cell, ability to produce the whole plant. That is called as totipotency. Right.
Yes, sir.
This totipotency character mainly localized with only meristematic tissues.
Yes.
Right, only mainly meristematic tissues. Based on this concept, we can get desirable plant. Based on totipotency concept, we can get desirable plants. Based on this totipotency, the tissue culture technique steps are involved in orderly. The first one, selection of explant. And during time of examination, if they ask about write about totipotency or totipotency step, or write about tissue culture technique steps, the both are same. Tissue culturing technique steps and totipotency steps both are same, and write about totipotency also same. If they ask write totipotency, just write the definition of totipotency and write the process of totipotency. The process involves the step, first of all, first step, selection of explant. Second, sterilization explant. Third,
Inoculation.
no, before that, selection, the preparation medium. Preparation medium. After that, sterilization medium. Now inoculation into the medium.
Medium.
Next, incubation. Next, transfer to the
Another medium.
Soil.
or first of all, not soil, first of all,
Nursery.
greenhouse.
Nursery.
Greenhouses. After next outfield.
Yes.
So they are the steps. This is very important.
We have discussed this earlier.
What, Parveen?
I think we have discussed earlier.
Completed is this?
According to my knowledge, it is completed.
Oh, What about others? Totipotency, concept, tissue culture, techniques completed or not?
No, sir.
Completed, sir.
Sure.
Sure.
Right.
Just explain in briefly, sir. I didn't hear.
Okay. For example, this is the outfield plant. First of all, let me clear the steps involved in this. First step, select.
And March 26 we have completed.
Let me complete in brief.
Yes, sir. I'm just
Select explant,
that's all.
sterilization explant, preparation medium, third, okay. Fourth one, sterilization medium, fifth, inoculation, incubation, transfer. So actually in thousands of plants in a crop field, there are thousands of plants are there, but among the thousands of plants, only one is high yielding and highly resistance power. For example, this is the highly resistant plant. We want get this type of plants more and more within a short time of period. For this, first of all, we selected the explant. What is the explant?
The plant which is selecting for tissue culture.
Not entire plant, any part of the plant.
Part of the plant, yes.
Any part such as the leaf or stem, root,
Yes.
anyone considered as a explant. Or anthers also, ovules also. Anthers, ovules, pollen grains, they can act as a explant. Now anyway, so where we collect the explant? From where?
From plant.
From plant should be outfield or in laboratory.
Outfield and healthy plant.
Right, outside healthy plant. So outside there is a chance to infected with many microorganisms, right?
Yes.
It should be
Sterilized.
sterilized. The sterilization with help of detergents, sodium chloride, sodium hypochlorite chemicals, and HgCl2 chemicals. With help of these chemicals, the explant should be sterilized. After that, second one, for growing explant, what are required? Not only growing explant, actually, what are the requirement for plant growth in general?
Plant nutrients.
Hormones,
soil,
nutrients,
Hormones,
like organic
nutrients,
nutrients.
and
Nutrients.
substrate also for fixation substrate also we can
Yes,
provide.
sir. Yes.
There we provide artificially inside the test tube the soil in the form of agar-agar to fixation to giving support,
Yeah, medium.
and some nutrients also we give provided, macronutrients, micronutrients, and hormones also provided. So they are medium. So the medium as it composition of micro and macronutrients, there is just infected with, there is a chance to infected with microorganisms, right?
Yes, sir.
That's why after preparation this medium, it should be sterilized in autoclave. In autoclave. What is autoclave? Autoclave produces the pressure, 120 degrees centigrade pressure. Due to this pressure and temperature, if any microorganism present inside the test tube, inside the nutrients, they are all will be killed. Right? So
Yes,
now
sir.
explant sterilized and medium also sterilized. What is the next step?
Inoculation.
Inoculation means introduce explant into the medium. Introduce explant into the medium, that is called inoculation. Of course, you sterilize the explant also, you sterilize the medium also. But during introduction, introduce explant into the test tube. Now there is a chance to infect it with microorganisms. If you do this process in outfield,
Yes, sir.
if you do this process outfield, exposed to the weather consisting so many microorganisms, so
Yeah,
easily
good
infected with
confirmation.
microorganisms. That's why this mechanism, this process, that means the inoculation process should be in closed, sterilized room. This sterilized room called as laminar airflow,
Yes.
right?
Yes, sir.
Now, in laminar airflow, the explant introduced into the test tube. Now it takes 2 or 3 weeks to observe the nutrient medium. Here we can observe here. Test tube, this is. After 2 to 3 days, the explant developed into callus like this. Explant developed into callus. Now after developed the callus, now you add some more hormones. The mainly some more phytohormones, mainly auxins and cytosine. Due to auxin cytosine ratio, the morphogenesis takes place. Morphogenesis means formation of shoot, leaf, and root.
Yes.
Shoot
Yes, sir.
leaf called as collagenesis, root formation called as rhizogenesis. That means from this region, from this callus,
Collagenesis means, sir?
embryo formation, sometime directly form the shoot. This is the direct. If it is indirect embryo formation, this embryo called as somatic embryo, and after this embryo developed into plant. Or in direct method, the callus directly produces the shoot. When we should produce the shoot after adding the phytohormones, auxins and cytosine. When you use auxin cytosine to this callus, from lower side rhizoids are produced, roots, and upper side leaf and stem are prepared, synthesized. The root formation called as rhizogenesis, stem formation collagenesis, rhizogenesis, and second one collagenesis. What happened finally after rhizogenesis, collagenesis? Small plantlets are formed. These small plantlets transferred into the
Greenhouse.
greenhouses. Greenhouse protect these tenure plants from the high temperature. After 3 to 4 weeks, they can grow some more, and now they transfer to the outfield. That's it. For example, here we can take 1,000 test tube, 1,000 explant. How many plants are produced?
1,000.
1,000 plants are produced from the single plant.
Yes.
Single plants, right? So
Within
this is
less
This is about totipotency concept or tissue culture technique. Clear?
time. Yes,
Yes,
sir.
sir.
Right. Now in the
Only
given
totipotency has capacity only. It is possible to plant cell only. No,
yes, plant cell only.
Yes.
no, not to animal cell.
Not animal cells. Already told you there are no meristematic issues,
Yes, sir. Yes, sir.
right? And that's why the lack of ability to produce.
Yes.
Next one.
Maybe
Stem cells, of course. Some stem
to
cells.
The regeneration capacity they may have.
create a hole.
From stem cells.
Yeah, stem cells and some of the animals they have regeneration capacity also, sir.
They are the non-chordates such as Coelenterata,
Yes.
Porifera,
Yes.
They are
Funny.
Leave
sponges.
that as
Sponges.
example. Sponges, yes.
Sponges mainly. Right.
Sorry, in the cloning technique, sir. In cloning technique, generally we are getting a whole organism also by taking a cell. So can we consider, sir, this is also a tissue culture? No, we are not growing. Just we are introducing into the animal body only again. In cloning also, we are first we are taking the zygote. That means cell ova collecting and another cell collecting. Nucleus are exchanging.
That is
A small doubt. Can we consider this as a what is this totipotency or not?
here totipotency. There are no fusion.
Okay.
Totipotency, no fusion. You can select any plant cell. We can regenerate into whole plants. Not a small portion.
Yes.
But as you said, in animals, you separately selected the gamete, male one and female gametes, both gametes you fertilized in a test tube.
That is
In a
in
test
vitro.
tube. No, yes, in vitro condition.
Yes,
In vitro conditions, after fertilization, zygote formed. This zygote developed into embryo. Embryo developed into baby.
Fetus. Yeah.
That is some different. There is no fertilization. How can we plant get in generally in conventional method? After fertilization, we can get the seed and germinate into plant.
Yes.
Not only plant, any organism, the same process. But in
The modern
totipotency process,
Chromosomal body.
right? In totipotency process, vegetative cell can produce number of plants. That's it.
Okay.
And totipotency concept. Should I explain these applications of tissue culture? Synthetic seed, status of micropropagation. Already explained. Already it was explained. But again, read yourself in just in brief. For example, what they given here? Applications of totipotency. The first one, production of synthetic seed. Synthetic seed. How the seeds are produced? Hello?
Yes, sir.
After fertilization.
Right. But in vitro condition, from explant developed callus, this callus indirectly developed into embryo. This embryo called as
Somatic embryo.
First of all, somatic embryo. The embryo should be protected with layer, isn't it?
Yes, sir.
Actually, in general, embryo. Where is the embryo in general?
Inside the
Right, ovule. The ovule is fertilized and developed into seed. That means embryo situated inside the seed and protected with help of seed coat, right?
Yes, sir.
But which one protect the somatic embryo?
We have to give the coating.
Yes, we will have to give coating encapsulated around it. This coating chemicals are polyxethylene and polyxyethylene and
This
sodium
actually this one
alginate.
we have not discussed.
Okay, polyxethylene and also sodium alginate and calcium alginate. Sodium
Calcium
alginate.
nitrate.
Yes, these are act as a encapsulated over the somatic embryos. Now these encapsulated membraned somatic embryos called as synthetic seeds. That's it.
Okay.
Is it clear?
Okay, sir.
Yes,
sir.
Right.
These
And
somatic embryos must be encapsulated
right.
with these chemicals.
Next, micropropagation. Propagation means
Distributed, increasing.
Cultivation.
propagate,
Distribute.
Number of
distribute.
copies.
Micro, it means within micro, the smallest explant can produce number of plants.
Yes.
The concept micro propagation, they get more and more number of copies, number of colors, number of embryos. That means number of plants are produced. Next one, no need to micro. These are all very short. These are not important one. Micro propagation, if it is, if they may ask in short answer. Synthetic seed, micro propagation, and also secondary metabolites also there, but not given here. Okay, don't worry. This micro propagation and secondary metabolites and synthetic seeds, they again repeated in practicals. I will explain there very clearly. Okay.
Okay, sir.
Right. Next, anther and pollen and ovule culture. Whatever it may be, anther or ovule, whatever it may be, same concept of tissue culture of totipotency technique, but here the change is only explained. Remember it. Only change is explained. For example, anther culture. What is the explant?
Anther.
Anther is the explant.
Ovule culture. What is the explant here?
Ovule.
Of course, here the given the name unnecessary, irrespectively unnecessary matter here. But don't get confusing about this matter. Just write about totipotency concept. For the anther culture and pollen culture and ovule culture same.
Okay, sir.
Oh, okay, sir.
Anther culture same, pollen culture same, and ovule culture same. There is small changes in applications. For example, what is the main concept of anther culture application? What type of plants we get?
Only male plants
Only
production.
male plants.
Right, and a diploid plant or applied plants?
Haploid.
Anther.
Only applied.
Anther.
Only haploid plants you will get.
Anther may get diploid also along with the applied. Anther means without form. Actually, in initial stage, inside the anther, microspore mother cell formed.
Yes, sir.
In that stage, we picked up the anther and cultured.
Okay, sir. Then
now the diploid plants are produced.
Okay.
Then what about the pollen grains? Pollen grains always applied.
Only haploid.
Only haploid plants are produced.
Okay.
And say ovule also same. Okay.
Only haploid female plants.
Right.
True study of particular trait.
Yes. Only applied plants we can get. About this pollen culture and ovule culture also. Methods of in vitro,
I
in
think
vitro.
with this we can clearly study the characteristic of the.
Right. We can clearly study the characters also. Okay, I will explain later this about all. Now another important topic is unit number 16, protoplast culture and somatic hybridization. Somatic hybridization, protoplast culture, somatic hybridization. So very easy and very important also for short, for long question. Right. Turn on page number
226.
Please be silent. Hello, please. 226, protoplast culture, somatic hybridization. Just wait. Right. So protoplast culture. So far we see the totipotency concept, and the same way the explant how to get into whole plant. The explant should be anther and ovule and pollen grains, whatever it may be, it can get whole plants. So now the protoplast. What is protoplast? Where it is?
Within the cell wall.
Right. You take the
I
cell.
mean, if we remove the cell wall, the remaining cell plant cell is called as protoplast.
Right. This is cell wall. This plasma membrane, protoplasm, without nucleus, cytoplasm. This, remember, this is cell wall, plasma membrane, protoplasm, the protoplasm with plasma membrane, these two together, plasma membrane, protoplasm, these two together called as protoplast. Cytoplasm only except nucleus. This except nucleus only, this is cytoplasm. What are present in cytoplasm? Hello?
Cell organelles are
Cell
there.
organelles.
All cell organelles present except the nucleus, right?
Mm.
Except the nucleus, all cell organelles are present. Okay? Right. Now let's see the protoplast culture. For protoplast culture, we should isolate the protoplast, right? In general, just assume how to isolate the protoplast. Before that, you should aware about the where the protoplast are existed. A protoplast existed just below the cell wall. If you remove the cell wall, we can isolate the protoplast, isn't it?
Yes, sir.
Right. Now, how can we remove cell wall? In our textbook, there are two methods. One is mechanical isolation and second one is enzymatic isolation.
Yeah.
First one, let's see the mechanical isolation. Mechanical isolation of cell wall.
Centrifugal method.
Mechanical method, the first one, osmosis method.
Okay.
In osmosis method, undamaged protoplast in strips are released by osmotic swelling. When it is occur when we placed in a low concentration of sucrose solution.
Yes.
Right. So for example, low sucrose content, cell, sucrose solution. The sucrose is low. Inside the cell, high content. What is the osmosis?
From low
moves.
concentration to high. The moment of
Right,
ions
no.
from the low concentration to high
Low concentration
concentration.
to high concentration. That's why from the low concentration from the beaker water moves towards the inside the cell. As a result, exit pressure falls on the wall, right? Exit pressure falls on the wall. To the exit pressure, it is swell and the over osmosis, the cell wall rupture. The cell wall somewhere the rupture. Here it is. Now remove the cell wall and we collect the protoplast. This is the one mechanical method, right? And another one is it clear?
Yes,
No?
sir.
Right. Next one, another one is with help of micro scissors. With help of micro scissors. This is done by cutting plasmolyzed tissue with sharp edged knife. The protoplast should be deplasmolysis. When it is deplasmolysis, when the cell deplasmolysis occur,
Kept in high concentrated solution.
right? Sweet. This high concentrated solution, for example, but inside the cell low, from the low concentration to high concentration, as we already told you. Now what will happen inside the cell? Actually, cell filled with, this is the cell, the closely attached with protoplast. That means from the plasma membrane. Plasma membrane, this is the cell wall. These both are fully attached, firmly attached with help of the full-fledged cell with help of full with water. When it is placed in high concentration, automatically this water moves out. Water moves out means what happened? Actually, this exit pressure due to the presence of water pressure, water content. But what happened later due to the when it is keep into high concentration, the inside the cell water moves out. As the water moves out, what happened? The pressure also decrease inside the cell, isn't it?
Yes, yes.
As a result, this plasma membrane separated from the cell wall. Now it's become like this, right? Is it clear? After remove of water, it becomes like this. Now under the microscope, specialized microscope, the watch this cell and cut with help of like this. Cut with help of sharp.
Microculture.
Now we can get the protoplast. This is to mechanical method, isn't it? It's okay? Hello? Right?
Okay, sir.
Mechanical isolation clear?
Yes,
Mechanical one is osmosis, another one is with help of the sharp edged knife.
yes, sir.
Next one, enzymatic isolation. Of course, in our textbook given the more matter about the enzymatic isolation, but I want to ask a very simple question to you. What are the chemicals of cell wall?
Cellulose, pectin,
Right,
suberin.
right. Suberin.
In some cases present, no, sir.
Okay.
Like in pericycle.
All right. Okay. Right, but
Lignin.
cellulose dissolved in which enzyme? Pectin dissolved in which enzyme?
Cellulase,
That's it.
pectinase.
Cellulase and pectinase. When the cells treated with cellulase and pectinase enzyme, automatically this cell wall this way. The cell wall dissolve. Cell wall dissolve the activity of enzyme. Cellulase enzyme,
Pectinase enzyme.
cellulase enzyme and pectinase enzyme. With help of cellulase enzyme and pectinase enzyme, due to this both enzyme, the cell wall dissolved. What is the rest portion except the cell wall?
Protoplast.
Now we can get the protoplast. Now the protoplast separated, right? By enzymatic
Yes,
method or mechanical method. After that, culture the protoplast. Culture the protoplast in a stages first. What they given in a textbooks. Not if that's so. Just wait. Right. Somehow we can isolate the protoplast by mechanical method or enzymatic method. Where it is protoplast cell wall regeneration. So here the protoplast separated by enzymatic or mechanical method. Correct. So anyway, this is one protoplast and this is another protoplast. Actually, you want two different plants because actually these two plants, one plant have the high yielding, another plant have the resistance power. This is high yielding power, this is resistance power. You want to get these two character within one plant. That's why you selected the two plant protoplast and cultured in petri dish. This is taken petri dish and agar agar, and this petri dish agar agar. These two protoplast mixed together. Now these both are mixed. This both protoplast mixed and both protoplast nucleus also mixed. And this is have some characters in the form of DNA. This also have some character in the form of DNA. Now these both are fused in test tube or in petri dish. Later, this is produced into callus tissue. After that, this callus, the process same add to the nutrient medium and preparation nutrients. What is their excellent? Hello?
Protoplast.
Here excellent is protoplast. And the same way the totipotency mechanism, same way preparation of nutrient medium, same micro macro hormones, and which produce the callus. After production of callus and had some more auxins and cytokinins hormones. As a result, due to the composition of auxin cytokinesis, morphogenesis takes place. Now the small plantlet form. Now this is transferred into outfit. That's it. Simple. Is it clear?
Yes, sir.
Right. And now see, let's see the agar medium preparation in incubation in small. No need this all. Sorry, agar. Okay. Right. If you understand the concept, you can easily understand about this culture of protoplast in our what they given in your textbook. Fusion of protoplast, somatic hybridization. Somatic hybridization, nothing but fusion of protoplast. Right. Chemical fusion. Okay, right. Electrofusion. Okay, right. No need. No need. Next one, hybrid. What is hybrid?
Soma-clonal hybrid.
Hybrids. Cytoplasmic hybrid. Hybrids, nothing but cytoplasmic hybrid. Protoplastic somatic hybridization or protoplast culture, same. Protoplast culture, somatic hybridization, same. But here the small difference between hybrids. Hybrids means cytoplasmic hybrid. Produce variable frequency in normal protoplast fusion experiment and contain nucleus one species but cytoplasm from both the parental species. Okay, right. This way. Cytoplasm. Hybrids.
Protoplasmic hybridization and somatic hybridization both are same, na sir?
Same. Both are same.
Okay, sir. Thank you, sir.
So here, after fusion of the protoplast,
Tell me.
Sir, after fusion of the protoplast, first we are taking two protoplasts here, so the nucleus get fused.
Yes, but not exactly. Nucleus may be fused or not. May be fused. If it is fused, it is called syncaryon condition. If it is not fused, it
Okay.
is called as heterokaryon condition. If it is syncaryon or heterokaryon, they can produce the both character.
Okay.
Right?
But the yes, okay.
In hybrids, without nucleus, only cytoplasm, right? That means in hybrid condition mainly, of course, here also not given clearly. XX medium methylation. This is cell A, cell B. Nucleus present. But here nucleus present, but it is inactive. Nucleus present, inactive. Now same here also protoplast separated with nucleus. Here also separated, but nucleus inactive. Now this both fused. In this situation, nucleus fusion or cytoplasm fusion takes place. Hello? That means it is sterile nucleus. Sterile nucleus means what is sterile?
Non-functioning.
Right, non-functional. The non-functional nucleus, non-functional nucleus, this non-functional nucleus can fuse with functional nucleus.
No,
Fuse?
may not be.
Can it fuse to the nucleus, functional nucleus? Hello?
No, sir.
No, only this nucleus with cytoplasm this is only cytoplasm, right? So the finally formation, this nucleus as it is, and this both cytoplasm present. Both cytoplasm, for example, this for example, yellow in color. This cytoplasm red in color. After fusion, red color and also yellow color, both are present. That means what would happen here? Hello? Cytoplasm fusion takes place. So this condition called as hybrids. This is called hybrid. So hybrid means only one active nucleus, one is fertile nucleus, another one is sterile nucleus. And here the given examples also, the page number 235, achievement of somatic hybridization. Somatic hybridization. The example given for the somatic hybridization protoplast culture, the best example given the Datura, Inoxia, Datura, Stromium. Datura, Inoxia, different species, Datura, Stromium also different. By fusing their diploid mesophyll protoplast. Mesophyll is the part of, hello? Leaf. Now the selected the leaf and separate the mesophills. From the mesophyll cells, the protoplast also separated. Now that these two protoplast in a agar medium, they fuse together. Now they can produce the only one plant. The produces the only one plant, and which is the both character acquired about the Inoxia and the Thrustromia, right? And another example is also given, Vareza sativa and Brassica nepos. Another example is also given here, but you can mention only any one enough. Vareza sativa, okay, right? So this is somatic hybridization achievement. Just wait. Only one is given. Somatic hybridization achievements. Only one is there. Somatic hybrids. Okay. Right, enough. One enough. Okay, right. And protoplast and hybrid. Only short question. Protoplast culture is a long question. These both are very, very important. And next
Okay.
one, transgenic plants. This is very interesting and very easy and very important. Transgenetic. What is transgenetic? What is transgenetic?
Transfer of genetic information. That means nucleus.
Right. So far, we can get the plants from the culturing.
Modification.
Culture from the almost related species. Now the transfer gene within the unrelated species. In tissue culture, only they selected only one plant. Right? Selected only one plant, or more than are two plants also, but these two plants related species. But transgenic means the transfer the genes from one genus to the another genus. That means one organism to the another organism.
Yes, sir.
Can you give any one example?
Bromato.
Transgenic plants example.
Bromato.
And BT genes, BT crops.
Yes.
BT crops. Do you know what is the BT?
Bacillus thuringiensis.
BT cotton. Bacillus thuringiensis.
Thuringiensis. Actually, this Bacillus thuringiensis kills the insect.
Yeah.
For example, the cotton. This is the. For example, this is the cotton plant. This most frequently this cotton plant infected with some more insect. Actually, the name of the insect also some gall insect, I think, like a caterpillar, like this. The insect like this. The mostly it is infected with this type of insect, right? Actually, this insect will be killed by Bacillus thuringiensis bacteria. Bacillus thuringiensis bacteria. Right? Hello?
Sir.
This caterpillar insect will be killed by Bacillus thuringiensis bacteria. That's why the scientists introduced this Bacillus thuringiensis bacteria genome. This is the Bacillus thuringiensis bacteria genome. This Bacillus thuringiensis bacteria genome separated from the bacteria and this genome inserted into the plant. Then what would happen? The plants also acquire the genome of the bacteria and acquire the character of the bacteria. What is the character of bacteria? The character of bacteria kills the insect. In the normal way, whenever the insect approached or reached this plant, cotton plant, when it eats, automatically the caterpillars would be died. This is the concept of transgenic plants.
Okay.
Transgenes. Is it clear? the concept of transgenic. Now we can observe when we are going to learn the methods, that is transgenic transfer of gene method, how the genes are transferred from the one organism to the another organism. What are the methods are involved in this? These
By
are all we can
Methods
observe.
needed.
gene therapy, genetic engineering.
So the genetic engineering methods, what are the genetic engineering methods? For this, we should know about genetic engineering techniques first of all.
Yes, sir.
Yes.
And after that, gene gun method,
Hmm.
micro injection method,
Hmm.
not the given. Okay. Of course, not given, but these are all important. We should learn these are all. Gene gun method, micro injection method, ballistic methods.
Hmm.
They are all very, very important. Okay.
Sir, given, sir. Gene gun given, genetic engineering given, sir. Next chapter.
Next step, where it is.
In page number 139, 239, sir. 238, 239.
Okay. Gene gun, okay, right. Micro injection,
Micro injection, not seeing, sir.
right. So we will complete later about these transgenic plants concept and methods and application. These three very, very important. Methods of gene transfer and transgenic applications, merits and demerits.
Okay.
Okay.
Okay,
Okay,
sir.
sir.
Thank you.
I want to ask some simple questions. Due to the BT, the beneficial to us or harmful to us?
Harmful to human beings, I think.
How it is harmful, sir?
Harmful or beneficial?
It is beneficial only, sir.
Harmful also, sir.
How, madam?
Harmful, and there also risk also there.
Uh-huh.
Remember the only this BT genes introduction only for commercial plants.
Okay, sir.
Only this
Yes.
is only for
For
commercial.
edible plants like the
Edible
brinjal,
plants,
potato,
right.
that is harmful to our health.
Harmful
Yes.
to our health. We will discuss later. These are all okay. Is it clear today, class?
Okay, sir. Clear, sir.
Right.
In first session.
Any doubts?
No doubt.
No, sir.
All of you.
No, sir.
No doubt.
Right. We will meet later. Thank you all.
Thank you, sir.
Thank you.
Thank you, sir.